Hello everyone, welcome to Doses Pathology Review, and we're going to start discussing cellular injury. And the first topic that we're going to discuss in cellular injury is cellular adaptations to stress. So let's get started. Cells normally maintain a steady state called homeostasis, and as cells encounter physiologic stresses or pathologic stimuli, they can undergo a process called cellular adaptation, and by undergoing adaptation, these cells achieve a new steady state and preserve their viability and function. However, if the adaptive capability is exceeded or if the external stress is inherently harmful, then cell injury is going to develop. This is going to be our map in discussing the cellular injury, and here we have a normal cell in a state of homeostasis. If this cell faces any type of stress, then it's going to adapt. If this cell is unable to adapt, then cellular injury will occur, and according to the severity of the injurious agent, the duration of the injury, and the type of injury, this injury can either be reversible or it can be irreversible. Reversibly injured cells can go back to normal with the removal of the stressful or the inciting agent. However, irreversibly injured cells are going to die, and they can die by one of two mechanisms. The first one is called necrosis and the second one is called apoptosis, and we're going to talk about these definitions later. But for now, we're going to discuss the first part of this map, and it is cellular adaptation. So, what are cellular adaptations? Adaptations are reversible changes in the number, the size, the phenotype, the metabolic activity, or functions of cells in response to changes in their environment. So, these changes can either occur together or they can occur one at a time. And the most important word in this definition, our keyword is reversible. So, all these changes are reversible changes. And these adaptations can either be physiologic adaptations or they can be pathologic adaptations. Physiologic adaptations usually represent responses of cells to normal stimulation by either hormones or by endogenous chemical mediators. An example of that is the enlargement of the breast and uterus during pregnancy. Pathologic adaptations are responses to stress that allow cells to modulate their structure and function, and by doing that, these cells escape injury. So from here, cellular adaptations are reversible, and these adaptations can be either physiologic or they can be pathologic. Cellular adaptations include hypertrophy, hyperplasia, atrophy, and metaplasia, and we're going to discuss all these cellular adaptations. So first, hypertrophy and hyperplasia. An increase in stress leads to an increase in organ size, and this increase in organ size occurs via an increase in the size of the cells and or increase in the number of cells. So increase in an organ size because of increase of the size of individual cells is called hypertrophy, and increase in organ size because of increase of the number of cells is called hyperplasia. And the most important thing about hypertrophy and hyperplasia that we have to know is the mechanism of both hypertrophy and hyperplasia. So hypertrophy involves gene activation, protein synthesis, and production of organelles. So our cells are lined by the cytoskeleton, and this cytoskeleton is formed predominantly of proteins. So in order to increase the size of the cell, you have to increase the size of the cytoskeleton, and you can do that by increasing protein synthesis. And whenever you're going to synthesize new proteins, then you're going to activate genes. In addition to that, we have to increase the production of the organelles inside the cells, and this is for the cell to maintain its function. So increased gene activation and protein synthesis makes the cell able to maintain its structure, and production of organelles will allow the cell to maintain its function. Hyperplasia, on the other hand, involves the production of new cells from stem cells. So we're going to synthesize new cells. Hypertrophy and hyperplasia generally occur together, and for example, the massive physiologic enlargement of the uterus during pregnancy occurs as a consequence of estrogen-stimulated smooth muscle hypertrophy and smooth muscle hyperplasia, increase of the size of the individual smooth muscle cells, and increase in the number of the smooth muscle cells. The tissues in our bodies are divided into three types according to their ability to proliferate. They are divided into labile cells, stable cells, and permanent cells. So first, let's discuss labile tissues. The labile tissues, or the continuously dividing tissues, are tissues in which cells are continuously being lost and replaced by maturation from stem cells and by proliferation of mature cells. And labile cells include multiple examples. For example, the hematopoietic stem cells in the bone marrow that are responsible for the production of the RBCs and white blood cells. And another example is the surface epithelium of multiple tissues in our bodies, like the skin, the oral cavity, and the intestine. Stable tissues are formed by stable cells, and these cells are quiescent and have only minimal replicative activity in their normal state. However, these cells are capable of proliferating in response to injury or loss of tissue mass. So, stable cells constitute the parenchyma of most other tissues, such as the liver, the kidney, and the pancreas. And with the exception of the liver, stable tissues have a limited capacity to regenerate after injury. The third type of tissue is permanent tissues, and the cells of these tissues are considered to be terminally differentiated and non-proliferative in postnatal lives. And permanent tissues include the cells of the cardiac muscle, skeletal muscle cells, and nerve cells, and the neurons. And these cells cannot make new cells, and because of that, they can never undergo hyperplasia, and they can only undergo hypertrophy. So we have the labile cells, the continuously dividing cells. We have the stable cells that do not usually proliferate, but in case of loss of an organ or tissue, they're going to start proliferating. And then we have the permanent tissues that cannot proliferate at all, and they are considered terminally differentiated. We have three examples for permanent tissues. We have cardiac muscle cells, we have skeletal muscle cells, and we have nerve cells or neurons. So, on the other hand, some cells, like the bone marrow stem cells that are responsible for the production of red blood cells and white blood cells, these cells undergo hyperplasia without hypertrophy. So the general rule here is that hyperplasia and hypertrophy occur together, but we have exceptions. Permanent tissues can undergo hypertrophy only, and bone marrow stem cells undergo hyperplasia only. So hyperplasia is divided into physiologic hyperplasia and pathologic hyperplasia. As we said earlier, these adaptations can either be physiologic or they can be pathologic. So we're going to discuss physiologic hyperplasia first, and it's further subdivided into hormonal hyperplasia and compensatory or reactive hyperplasia. Hormonal hyperplasia is exemplified by the proliferation of the female breast at puberty and during pregnancy. And compensatory hyperplasia or reactive hyperplasia is the type of hyperplasia in which residual tissue grows after removal or loss of part of an organ. And a clear example for that is when a part of a liver is resected, then the remaining cells of the liver are going to begin proliferating as early as 12 hours later, and eventually these cells have the ability to restore the liver to its normal weight in just a period of 4 to 6 weeks. So this is an example of compensatory or reactive hyperplasia. Regarding pathologic hyperplasia, is the type of hyperplasia that is usually caused by excessive hormonal stimulation or by excessive growth factor stimulation. And an example of pathologic cellular hyperplasia is endometrial hyperplasia. So the endometrium is usually stimulated by the action of estrogen. So estrogen can cause increase in the number of the smooth muscle cells of the uterus, specifically of the endometrium. However, if this estrogen is excessively secreted, then what's going to happen is pathologic endometrial hyperplasia, and pathologic endometrial hyperplasia can progress to endometrial dysplasia, and eventually this can progress to endometrial cancer. So the rule is pathologic hyperplasia can progress to dysplasia, and this dysplasia can progress to cancer. A notable exception for this rule is benign prostatic hyperplasia. In BPH or benign prostatic hyperplasia, there is no increased risk for prostate cancer. So this is hyperplasia. It can be physiologic hyperplasia or pathologic hyperplasia. Physiologic hyperplasia can be hormonal, or it can be compensatory or reactive hyperplasia. Pathologic hyperplasia can progress to dysplasia, and dysplasia can progress to cancer. Now, we're going to talk about hypertrophy, and hypertrophy can be physiologic hypertrophy or pathologic hypertrophy. Example of physiologic hypertrophy is the enlargement of the smooth muscle cells of the uterus during pregnancy, and an example for pathologic hypertrophy is the enlargement of the heart that occurs in people with systemic hypertension. Now we're going to discuss atrophy. So atrophy is the opposite of hypertrophy. So in hypertrophy, there is an increase in the size of an organ because of increase in the size of cells. And in atrophy, there is a decrease or shrinkage in the size of an organ because of shrinkage or decrease in the size of cell. And this occurs by the loss of cellular substance. And there are multiple causes of atrophy. These causes include decreased workload, loss of innervation, decreased blood supply, inadequate nutrition, loss of endocrine stimulation, and aging, which is called senile atrophy, like the brain atrophy that occurs in old people. And you don't actually have to memorize all these causes. You just have to understand that these causes can result in cellular atrophy. In atrophy, there is decreased cell number and decreased cell size. And decreased cell number occurs by apoptosis. Apoptosis is programmed cell death or cellular suicide, and we're going to discuss apoptosis in another video. But for now, I want you to know that decreased cell number in atrophy occurs via apoptosis. Decreased cellular size can occur by two ways. The first one is decreased protein synthesis, and this occurs because of decreased metabolic activity of the cells. And the second way by which cell size decreases is increased protein degradation. So as we said earlier, the cells are lined by the cytoskeleton, and in hypertrophy, we said that in order to increase the size of the cell, then you have to increase the size of the cytoskeleton and you have to produce new organelles. In atrophy, the opposite will happen. So the size of the cytoskeleton will get smaller and we have to degrade and get rid of some of the cellular organelles. So the two ways by which these changes occur are ubiquitin proteasome degradation pathway and autophagy. So what is the ubiquitin proteasome degradation pathway? So the proteasomes are cellular components or cellular organelles that are responsible for digestion of unwanted proteins, and these proteasomes can recognize unwanted proteins when these proteins are tagged with ubiquitin. So the intermediate filaments of the cytoskeleton will be tagged by ubiquitin, and the proteasome will recognize these intermediate filaments as unwanted, and then it's going to digest the proteins of the cytoskeleton, and this is going to result in decrease in the size of the cytoskeleton. And the second thing is autophagy. So autophagy means self eating. So the cell is going to eat its own components, and it does that by generation of autophagic vacuoles that contain organelles. These vacuoles fuse with the lysosomes that contain hydrolytic enzymes, and these lysosomes are going to destroy the organelles and the substances that are found inside these autophagic vacuoles. So let's say that again. Atrophy, decreased cell size and decreased cell number. Decreased cell number occurs by apoptosis, and decreased cell size occur by decreased protein synthesis and increased protein degradation. Decreased protein synthesis occurs because of reduced metabolic activity of the cells, and increased protein degradation occurs by the ubiquitin proteasome degradation pathway and by autophagy or cellular self-eating. The last adaptation process that we're going to discuss is metaplasia. And metaplasia is defined as a reversible change in which one adult cell type is replaced by another adult cell type, and it most commonly involves changes of one type of surface epithelium to another type of surface epithelium. However, it can occur in epithelial cells and it can occur in mesenchymal cells as well. And we have three types of surface epithelium in our bodies, and these types are squamous epithelium, columnar epithelium, and urothelium or transitional epithelium that lines the urinary tract. So why does metaplasia occur? Metaplasia occurs because the metaplastic cells are better able to handle the new stress. The classic example for metaplasia, and the one that is commonly tested, is Barrett esophagus. So what is Barrett esophagus? Remember that we have the esophagus, which is lined by stratified non-keratinizing squamous epithelium, and we have the stomach, which is lined by non-ciliated mucinous producing columnar cells. So in Barrett esophagus is a condition we see in people with gastroesophageal reflux disease. So the stomach is lined by the non-ciliated columnar mucinous epithelium because this epithelium have the ability to withstand the acidity of the stomach, and the esophagus is lined by the non-keratinizing squamous epithelium because this epithelium is suited to handle the friction of food and chemicals. So what happens here is that the acid reflux from the stomach will cause irritation in the lower part of the esophagus, and because of this irritation, the cells of the lower part of the esophagus will undergo metaplasia from non-keratinizing squamous epithelium into non-ciliated mucinous columnar epithelium because this type of epithelium is better able to handle the acidity of the stomach. Another classic example is the squamous change that occurs in the respiratory epithelium of habitual cigarette smokers. So the trachea and bronchi are lined by ciliated columnar epithelial cells. However, these cells cannot handle the stress that is caused by the chemicals that are present in tobacco. So the metaplasia occur, and these cells transform from ciliated columnar epithelial cells into stratified squamous epithelial cells, and the stratified squamous epithelial cells can withstand the chemicals that are present in tobacco. So now we're going to discuss the mechanism of metaplasia, and metaplasia occurs via reprogramming of stem cells which then produce the new cell type. So in metaplasia, it's not merely changing how the cell looks, the morphology of the cell. It's by reprogramming of the stem cells to produce the new cell type that we need to combat this stress. And metaplasia is like every other cellular adaptation mechanism, is a reversible mechanism with removal of the driving stressor. For example, treatment of the gastroesophageal reflux by antacids may reverse Barrett esophagus, and the cells can go back to normal. Okay, although the process of metaplasia is not in itself pre-malignant, metaplasia is sometimes associated with progression to malignancy. So Under persistent stress, metaplasia can progress to dysplasia and eventually result in cancer. And an example for that is Barrett esophagus, which may progress to adenocarcinoma of the esophagus. So the condition here is like the pathologic hyperplasia. Pathologic hyperplasia can progress to dysplasia and then it can progress to cancer. And here is the same. Metaplasia can progress to dysplasia and dysplasia can progress to cancer. And the classic example for that is Barrett esophagus that may progress to adenocarcinoma of the lower part of the esophagus. However, this is the general rule, but we have an exception, and this exception is apocrine metaplasia of the breast. It's a type of metaplasia that occur in breast cells, and we're going to discuss this later. However, for now, I want you to know that apocrine metaplasia of the breast carries no increased risk for cancer. So in hyperplasia, benign prostatic hyperplasia carries no increased risk for cancer, and in metaplasia, apocrine metaplasia of the breast carries no increased risk for cancer. Another point worth mentioning is that vitamin A deficiency can also result in metaplasia. So the thing is, vitamin A is necessary for the differentiation of specialized epithelial surfaces such as the conjunctiva covering the eye. The conjunctiva is a thin, delicate membrane composed of thin squamous cells, and they line our eyes. In vitamin A deficiency, this epithelium undergoes metaplasia from thin squamous epithelium into stratified keratinizing squamous epithelium, and this can increase the risk for blindness, and this change is called keratomalacia. So the message here is that vitamin A deficiency can also result in metaplasia. As I said earlier, metaplasia commonly occurs in epithelial cells. However, mesenchymal or connective tissue can also undergo metaplasia, and a classic example is myositis ossificans, in which muscle tissues changes to bone during healing after trauma. So myositis ossificans, in this condition, when muscle cells are subjected to trauma, they're going to undergo inflammation, and that is why it's called myositis. Myositis means inflammation of the muscle. So what happens here is that inflammation occurs, and because of inflammation, the muscle cells will undergo metaplasia and be transformed from muscle cells into bone cells, and that's why it's called ossificans. And this is an example of metaplasia in mesenchymal or connective tissue. So here we're done with cellular adaptations. We talked about hypertrophy, hyperplasia, atrophy, and metaplasia. And for now, we're going to discuss a couple of changes that are not considered a part of cellular adaptations. However, they are really important to understand. The first one is dysplasia. Dysplasia is abnormal or disordered cellular growth, and that is how I remember it. Dysplasia, disordered cellular growth, and it most often refers to proliferation of precancerous cells. And we already discussed a couple of things about dysplasia. It often arises from long-standing pathologic hyperplasia or metaplasia. For example, long-standing endometrial hyperplasia can progress to endometrial dysplasia, and endometrial dysplasia can progress to endometrial cancer. Barrett esophagus is a type of metaplasia, and we said that metaplasia can progress to dysplasia and dysplasia can progress to cancer. However, dysplasia is similar to metaplasia and to the other cellular adaptation mechanisms in the sense that it is a reversible change with the alleviation of the inciting stress. However, if this stress persists, then dysplasia is going to progress to carcinoma, and when it progresses to carcinoma, then this is an irreversible change. Okay, so now lastly we're going to talk about aplasia and hypoplasia. Aplasia is a failure of cell production, and it can either occur during fetal development or later in life. Okay, During fetal development, aplasia results in agenesis or a complete absence of an organ due to failure of production. And classic example is unilateral renal agenesis. And patients with unilateral renal agenesis have only one kidney, and they usually present later in life. So someone can ask, how can one function with only one kidney? And the thing is, the kidney that is present is going to undergo cellular hypertrophy and is going to work the work of two kidneys. However, when this stress is excessive, then cellular injury is going to develop, and these patients usually present with renal failure. That is how they are discovered. Okay. This is regarding agenesis. Later in life, aplasia can occur as well, and it can be caused by permanent loss of precursor cells in proliferative tissue, such as the bone marrow. We have a virus that is called B19 virus, and this virus can damage the hematopoietic stem cells that are present in the bone marrow, and it can cause a condition called bone marrow aplasia. So this is aplasia. Hypoplasia is a decrease in cell production during embryogenesis, and it results in a relatively small organ like the streak ovary in Turner syndrome. So now we finished the cellular adaptations, and in the next video we're going to talk about cell injury. Good luck and see you in the next video.